Hello, everyone. I would like to welcome you to today's webinar on the Human Somatic Mutation Database, or HSMD, Harnessing Insight from Real-World Oncology Cases and QIAGEN Knowledge to Annotate Variants and Assess Prevalence, Introducing a New Somatic Database, Our Best Kept Secret. This will be presented by Dr. Beate Litzenburger. My name is Mary Ann, and I'll be the host and moderator for today's session. During today's webinar, all attendees will be in listen-only mode to avoid any background noise during the presentation. As for our webinar platform, you'll see several windows open in your browser. There is a media player window that will be showing our presentation. Depending on the size of your screen, I would encourage you to play around with the sizes of the windows if anything is unclear or too small. You can minimize or maximize using the icons in the top right of the windows, or you can click and drag on the edges. There's also a Q&A section called Ask Question. If you have any questions for the presenter or regarding our solutions in general, please let us know here. We'll take some time to address your questions during a Q&A session at the end of our presentation. If there are any questions we don't get to today, we will follow up with you by email. You'll also see several other possibilities to open other windows at the bottom of your screen. If you click these icons, the windows will open, and when you click again, they'll disappear. So now I would like to introduce our speaker for today, Dr. Beate Litzenburger. Beate is a Global Product Director of Oncology for Clinical Decision Support Software and Interpretation Services at QIAGEN. Prior to joining the QIAGEN team, Dr. Litzenburger led clinical decision support and the interpretation of patient cases for several next-generation sequencing protocols at the MD Anderson Cancer Center. In this role, Dr. Litzenburger was responsible for providing alteration interpretation, drug, and clinical trial options to clinicians. Now I will hand the presentation over to Beate.
Thank you so much for the nice introduction, and thank you all for attending today. QIAGEN's curated knowledge can be used for many purposes. However, we have particularly deep curation in the area of cancer research. As you know, cancer is a disease caused by mutations. While the majority of cancer is caused by spontaneous or somatic mutations, many cancer-relevant or cancer-associated variants can be inherited. To understand the complexity of cancer genetics and the oncogenic potential of a mutation, a resource is needed that covers both types of mutations, and HSMD fulfills that need. Datasets such as TCGA and ICGC provide opportunities to identify causative variants underlying human cancers. However, most of the somatic mutations do not have a noticeable effect and are so-called VOSs. And the prevalence of VOSs in a cancer sample hamper the distinguishing of cancer-causing driver mutations. Many approaches and databases have been developed to prioritize cancer-driving mutations. Most of these tools can be classified into three categories based on three basic principles. Frequency-based methods, which consist of identifying mutations that are more frequently mutated than the background mutation rate. While this method is an essential step at identifying driver mutations, as a standalone measure, it's not sufficient and can be confounded by the fact that mutations can generally occur with a high frequency in the general population. The second principle is based on computational algorithms that have been developed to predict whether a mutation is deleterious or pathogenic based on concepts including evolutionary conservation, structural constraints, and the physiochemical attributes of amino acids. But cancer driver mutations predicted by these computational methods lack consistencies and are prone to false positives or negatives. The third method is purely manual research by subject matter experts, sifting through the literature to find publications that describe statistically powered experiments that decipher the biological, clinical, or disease relevance of a mutation. This is an unsustainable, laborious effort, and it's not scalable. Over the last years, many tools such as OncoKB, My Cancer Genome, or the Jackson's Lab knowledge base have been developed to deposit curated variants with evidence-based information about individual somatic mutations into a database. However, they have limited number of variants, some of them less than 10,000 mutations that have been annotated towards its biological, clinical, or disease relevance. Now, as a scientist, keeping the pace with new discoveries is a serious bottleneck. I hope this picture literally speaks to the problem that we face as scientists, and is that its valuable information can go really undetected due to inadequate data mining and variant interpretation mechanisms. While mutation and gene prevalence remain one of the measures to identify driver mutation, it is important to use different genomic oncology data sets to calculate frequency data depending on the goal of your research study. HSMD is powered by QIAGEN's aggregated knowledge. Since more than 20 years, QIAGEN's subject matter experts and ontology engineers have been using sophisticated curation tools and ontologies to transform unstructured genomic and cancer-related literature into machine-readable units with semantic consistencies. The aggregated structured genomic and cancer-related knowledge comprises of biological, phenotypic, therapeutic, and outcome data that can be used to understand the genetics of a disease, review available therapeutic options, decide on experimental strategy, or use this data as a part of accelerating a drug development program. The Kaijun knowledge base model allows us to answer unexpected questions because the database can transverse indirect relationships, build new relationships, and can find unexpected connections. For simplicity, the most relevant interconnected aspects that can be explored in HSMD are shown here. This dataset is genomically described by RefSeq and Ensemble transcripts and is currently available in Genome Build 37. The clinical oncology dataset comprises of over 300,000 de-identified patient cases, and we capture age and gender of the patient. The interconnection between the variant, tumor type, and investigational and approved drugs allows the user to explore therapeutic relations. The mutation is characterized by 4 measures: the actionability classification according to the AMP guidelines, an activation type, molecular function summary, population impact, and biochemical impact. All relationships are backed up by references for the user to transparently review the underlying evidence. So what are the sources that establish those relationships, and how many relationships can be assessed through HSMD? As of today, HSMD contains over 3 million curated alteration findings, and this number goes up during weekly updates of the content. The data within HSMD is derived from a combination of information from the Kaijun knowledge base, observed clinical cases, as well as public databases used to annotate variants. These resources are combined to bring a wealth of usable, trustworthy information regarding the biological, clinical, and disease relevance of an alteration to view. Again, this information comes with appropriate citations for transparency. Currently, the curated data characterizes over 1 million unique variants encompassing 1,400 different genes and nearly 11,000 cancer types and subtypes, as well as hereditary cancer syndromes. Now, I would like to spend a minute on describing more details about the oncology dataset, patient dataset that is incorporated into HSMD. For more than a decade, Kaijun's professional interpretation team, consisting of Ph.D. level scientists, as well as a team of collaborating oncologists, has been researching and interpreting somatic variants reported in cancer patients. This clinically observed NGS data is derived from a number of clinical testing labs using a variety of different sequencing panels. And the manual variant interpretation includes the activation type, the actionability according to the AMP guidelines, and the range of tumor types include solid tumors, lymphomas, and hematological cancers. The oncology dataset is constantly growing and will add about 70,000 new cancer profiles annually into HSMD, including 60,000 new curated variants. Within oncology, a number of different aggregators, publicly or privately, have emerged, and each of the datasets has its own strengths and limitations. Some datasets may be larger than others, but lack a biological or clinical annotation. Other datasets may be biased towards a certain disease. So in some cases, combining datasets together can reveal novel insights. As an example, how does HSMD dataset differentiate from TCGA? First of all, it's important to recognize that the Kaijun dataset has a larger sample size. It is more diverse and it reflects a patient population that tend to be post-treatment. The TCGA dataset contains a huge number of variants, noticeably due to VOSs and polymorphisms. Whereas in contrast, HSMD dataset contains more than 260,000 unique variants enriched for presumed clinically relevant mutations. So if you ask the question, what are potential acquired resistance mutations that may occur due to extensive treatment, you will likely not find the answer using TCGA, where the dominant samples were derived from treatment-naive cancer patients. What I'm trying to illustrate is that it's important to recognize the characteristics of each dataset to be able to use each of these datasets optimally to answer your scientific questions. Now, this graphic represents the number of cases tested for each of the genes, comparing HSMD's dataset in blue to COSMIC's sample coverage from their targeted screen data, which is red. HSMD contains a large number of cases tested for each gene, providing a mean for a robust and confident assessment of gene and variant prevalence from clinical derived cancer cases. So how can you access HSMD? In its first version, the Kaijun oncology dataset can be accessed via a searchable interface. The interface allows for flexible searches by a specific term, such as a variant, gene, drug, disease, or searches across the dataset. So let's dive into the application, and within the next 10 to 15 minutes, I will walk you through some of the many use cases HSMD can answer. So this screenshot here represents an interface of our Gene Explorer, which allows for exploring and prioritizing a list of genes with specific attributes that are listed within each column. The colored bars visualize the overall genetic characteristics of a gene based on the curated mutations within each gene shown in the third column. For example, the function column in the middle shows the proportion of mutations in each gene which contain known functional impact classifications. Hovering over the colored bars will show the details of the distribution that make up the genetic characteristics of a gene. All data in HSMD is hyperlinked for you to dive deeper into a gene-bearing drug, trial, or disease and respective references. Now, what can you do with this information? If an oncogene or tumor suppressor status is not established, one can predict a potential status based on the general composition of the alteration types, activation status, and tier classification. One can filter and sort this data set to prioritize a list of genes that are known tumor suppressors or oncogenes with known functional impactful mutations that are clinically relevant. One can derive a list of genes that contain mutations that cause splicing defects, or prioritize based on known functional impact classifications. And one can ask questions around which gene on my panel are actionable, or which oncogene are targeted with the most or least drugs. The Variant Explorer allows you to explore and prioritize the list of variants with specific attributes. So each variant is described with its impact, function, actionability, with how many diseases it's associated with, and how many associations from the literature are available for each variant. Now, the column allows you to identify if we have seen this variant in our real-world oncology data set called clinically observed, or if this is derived from our Kaijun knowledge base through proactive curation. One can interrogate the variants by asking the questions such as, what are the missense mutations with a known biological and clinical relevance that have been clinically observed? Or which biological relevant mutations have associated therapies or clinical trials? Or which biological relevant variants are emerging biomarkers without associated therapies? Which variants have been most or least characterized in the literature? And among those variants that are bioassays, which ones are associated with a lot of literature? Now, the Alteration Explorer also provides the means to search on a codon position to identify how many variants we have observed at that location, how were they classified, and if we have seen them in our clinical data set or in our Kaijun knowledge base by proactively curating the literature. You can explore HSMD from a disease, general disease perspective. For example, you could ask questions, which diseases were clinically observed and have TERT and EGFR frequently mutated? The data can also be filtered towards which cancer types have we observed in our clinical data set, but are not associated with clinical trials or drugs. Or what are some of the diseases that have been associated with the most or least variants? Now, I will take an example of a gene, Btk, to walk you through the specific gene and variant content in HSMD. First of all, the left side contains headers that serve as navigation pane and allows you to easily and quickly navigate and jump from section to section through the gene or variant specific content. The gene summary allows you to quickly understand if function or if to understand its function and if somatic or germline mutations have been associated with a hereditary syndrome or solid or hematological malignancies. Various link outs exist to other databases such as IPA, to understand in which pathway the gene is located. The subsequent sections provide an overview of the alteration types and functional impact distribution within this gene. You can easily find out what are the known activating mutations by clicking on the number, which navigates you to the alteration section, which then displays those specific 9 mutations that are known to be activating. Alternatively, you can ask the questions, what kind of alteration types are frequently observed, such as indels, SNVs in these genes? And among a specific alteration type, in this example, missense mutations, how many are known to be activating or predicted activating? Again, the alteration section will automatically display just missense mutation, and filtering capabilities allow you to select only activating or predicted activating mutations. But what are really the affected codons in a gene of interest at which mutations occur? Are there any hotspots? HSMD displays a lollipop graphic that allows you to identify where in the gene and how often we have observed clinical cases with a specific mutation at a codon. And hovering over the lollipop, you can determine the amino acid position for this hotspot. Now, you may want to review in which disease is BTK predominantly mutated. In this example, CLL and CLL-SLL is shown up as the top disease frequently mutated. If you don't see your disease in the top 10 on the screen, you can either use the arrows to navigate through those 34 diseases in which QIAGEN has observed this gene to be mutated in. Alternatively, you can use the search box or sort the table by the presented columns to answer additional questions, such as how many cases in each disease were tested for this gene. Now, for a particular disease of interest, such as CLL, it is in general important to understand the mutational landscape in this disease. So you can hyperlink to that disease and navigate to a disease page, which provides detailed information about the disease in several sections shown here on the left. The gene prevalence in this particular disease is described in the gene distribution section, allowing you to understand the top mutated genes, alteration as well as alteration types, and the top reported variants in this disease. Now, from this graphic, you can determine that BTK is actually not one of the top mutated genes in CLL. So you may be wondering, how actionable is this gene? And what are really the most clinically significant mutations within this gene? HSMD provides for each variant in actionability classification based on the AMP guidelines, which determines the therapeutic, prognostic, or diagnostic importance of a variant. We can then plot the actionability across the different alteration types in the gene. This provides an overall assessment how actionable a gene is, if it's rather an emerging gene, or if the actionability is driven by a small number or large number of variants in a specific alteration type. Now, there are 9 mutations that stand out to be clinically relevant. If you click on this, these 9 mutations occur all at the same codon that we have previously identified as a hotspot in this gene. Now, if those variants are clinically relevant, are there any targeted are they targeted by a drug or cause resistance to a drug? And the drug section provides a list of drugs, its target, and a therapeutic response related to a specific disease. The information you will find in the case of BTK is that there are 3 drugs that are approved by an agency, acalabrutinib, ibrutinib, and zanubrutinib. However, the specific hotspot at codon 481 causes resistance to BTK inhibitors identified in a variety of diseases. I mentioned that each relationship is associated with references, and if you use the drop-down next to each drug, you will find the relevant papers and a summary statement to support this relationship. So what is known about this particular variant, and what is the biological impact, and how frequently has a specific variant been observed? So for each mutation, HSMD organized detailed information about a variant into several sections shown here on the left. The description is an expertly written summary of the molecular function backed up with hyperlinks references for your review. In the case of this BTK mutation that causes resistance, you can find out that the C41 residue is actually a binding site for the BTK inhibitors, and if mutated, decreases the binding and affinity of BTK inhibitors. HSMD also displays the detailed genomic description where this mutation is located, its nomenclature based on the different RF-seq and ensemble transcripts. Now, alternatively, HSMD allows you to confidently distinguish variants of uncertain significance from those that may have clinical relevance. Our variant scientists did a targeted curation for this variant and specifically researched the literature and database sources and determined an unknown impact. Now, many cancer-associated variants have been primarily studied in a hereditary cancer or inherited disease context. HSMD provides comprehensive literature coverage about a variant, including literature related to hereditary cancers or inherited diseases that could shed a light into a potential role in cancer. In this example, we have clinically observed this BTK mutation. However, a role in cancer has not been established yet, but has rather been associated with X-linked agammaglobulinemia. If you are not really familiar with this specific hereditary disorder, you can go and jump to the bibliography, search for this specific syndrome, and hypothesize a potential role of this mutation in tumorigenesis by reviewing papers, especially functional study papers, that shed a light into its molecular function role and potentially subsequently a role in tumorigenesis. Now, going back to the example of the C481 hotspot again, we can also ask the question, what is the most prevalent disease at which this mutation has been observed? Data can be reviewed in the observed clinical case distribution section, and additional disease associations can be derived by searching through the bibliography to find additional relationships. So how does HSMD characterize rare variants with early clinical and or preclinical actionability information? So this is a tier 2 variant, which means it's a potentially clinically relevant mutation. It is a known activating mutation, and the description typically describes early results or case studies with a potential response. The fact that it's rare is supported by the frequency calculation from our data set. Now, within the drug section, case studies are captured that describe the treatment outcome of a specific cancer type harboring this mutation. However, for this mutation, it is not established that the mutation sensitizes the tumor to the drug. But when you're looking at investigations and investigational drugs, this is exactly being tested in clinical trials for lymphomas, plasma cell myelomas, and solid tumors. Now, HSMD can link directly to external databases such as HGMD, ClinVer, DBSNP, OncoKB, and also incorporates population frequency data from NOMAD. And the NOMAD frequency data can specifically and transparently be reviewed to determine if a polymorphism is predominantly present in a specific subpopulation. Likewise, the variant page displays a variety of prediction tools which may be used as supporting evidence to evaluate the biological role of a mutation. HSMD contains a clinical trial portal that allows you to search globally clinical trials based on specific search criteria, such as the gene, the phase and the recruitment status, a specific drug, and then country, city, and arranged around the city. Now, each trial is hyperlinked, and within each trial record, it describes the specific trial details, its location, the eligible alterations, drug and disease. And then you can also further go into deeper evaluation. If you want to understand the specific drug targets and its development status of this type of drug, you can go to, you can link to the drug page, which then it can display approved brand names, synonyms, and any relevant drug targets. So in summary, HSMD contains a comprehensive oncology data set that gives you access to a rapidly growing real-world oncology data set and structured curated evidence. The content is updated weekly to cope with the increasing number of variants, literature sources, and changing clinical relevance. The first version of HSMD is developed in an easily accessible user interface. Now, I hope that with the use cases today, I could show that HSMD allows you to spot key mutations with driving properties or clinical relevance, that the comprehensive aggregation of structural content can bring valuable scientific information to your attention that can be used to accelerate new discoveries or drug development programs and to interpret confidently clinical relevant variants. Kaijun has been curating and organizing clinical data for decades, and we would be very happy to be your partner by providing access to our high quality, trustworthy, updated, and comprehensive content in a way that suits your individual needs best. Please visit our websites to request a free free trial access. HSMD is a free trial access for 5 days. Please sign up, and then thank you for your attention and time today.
Thank you, Beate, for that informative presentation. So we'll now use the rest of the time to answer any questions that you may have. Please use the Q&A panel to submit your questions for our speaker. The first question that we have today is, have you calculated co-occurring genes and variants, and is that data available in HSMD?
So we have done internally our calculations on co-occurring genes. However, we have not yet implemented the results of this analysis within HSMD, and we're expecting to do this over the next year.
Okay, thank you. The next question that we have is, how often is this database updated?
Yeah, I mentioned that during my presentation already, but it's good to repeat. HSMD's variant curation content is updated weekly. The clinical data set externally incorporate databases such as NOMAD, HGMD, etc., updated quarterly. And then within HSMD, we do expect to add about 5,000 new curated variants per month and about 70,000 clinical cases annually.
Okay. Another question that we have is, will this data be available in GR38?
Yeah, that's a good question. The initial version is built in Genome Build 37, and we plan to lift it over to GR38 next year.
Okay. Another question on data is, will this data set be available as a download?
So our plans are to expose our content through the API next year. If people need early access, we do have a solution through our services for selected customers. So if there's an interest in an API access or a customized solution, please reach out to our professional service team at Kaijun.
Great, thank you. Okay, to follow up on an earlier question, you mentioned that HSMD is updated weekly. How do you ensure that the content is up to date?
Yeah, that's a good question. So our content curation team has nightly sophisticated queries that run on PubMed, guidelines, and labels. And this identifies a set of new articles and other references, which are then further processed through a screening process, and which may eventually end up in the task list of curators who then characterize the articles and place any additional information into a structured format based on the appropriate curation protocols. The variants that have been clinically observed at least twice over the last year will undergo a review process to gather the latest evidence and to decide if the activation type, molecular function summary, and actionability is up to date.
Okay, great. Okay, the next question is, do you use ML and crowdsource data for variant curation?
That's an interesting question. So the curation of data at scale is certainly a very complex process, and Kaijun uses various methods whenever it's appropriate without compromising the precision. So the process of clinical and phenotypic data curation is laborious, intellectually intensive activity, and it demands a deep subject matter expertise and significant personal resources. However, it is important to recognize that using the most appropriate method at each step in the curation process is critical for operating at scale and coping with the growing body of literature and databases. So, for example, the process of identifying and quickly prioritizing relevant publications for curation is commonly carried out by querying articles with computer programs. Some of the manual screening the content for its applicability. And in this case, machine learning is actually providing some valuable approach to enhance and accelerate this process. There are other methods like the natural language processing and certain machine learning approaches can further facilitate the seeding of relationships. So from articles in order to describe, for example, a phenotypic or genotypic relationship. And this really helps to focus our human curators or domain experts to spend most of the time to focus on some of the challenging tasks that machine learning and other algorithms have not yet tackled with high precision. And that's typically precisely and organizing information relationships for clinical studies, capturing really the complex relationships of interactions between variants and the complex clinical outcome data, including graphics, supplementary material. I think this is still not very well refined using computational methods. And for us, the high-touch human review process is still the cornerstone and of our curation to ensure high accuracy and high specificity and precision and also relevance for that specific mutation or disease.
Okay, got it. So it looks like we'll still have time for a few more questions. So if you do have any questions for Beate, please go ahead and submit them. If we don't get to your questions today, we will follow up with you by email. The next question that we have is, when will HSMD be available, and is there a way to try it out?
So HSMD is available already. It was launched in November, and so you can try it out by requesting a trial access first. But please feel free to reach out to also our specialties sales that allows you to get more information about HSMD and have a conversation with them as well. But it is available already for you to try it out.
Okay, thank you. And if anyone is interested in more information, on the screen, you can see our website is www.digitalinsights.QIAGEN.com. You can find more information on HSMD there, and you'll also be able to request a trial. Okay, the next question that we have is, will you be able to overlay the content with patient data such as age? For example, to review mutations in pediatric patients?
And so yes, we certainly do have these capabilities to do that. However, it is not part of the first release, and we'll have to decide if it's if it's part of the subsequent release.
Okay, I think we have time for one last question. Our final one is, you showed examples of just variants. Does HSMD include fusions or CNVs?
Yeah, currently the interface only displays small mutations. However, that's part of the next release. We do plan to add fusions and copy numbers.
Okay, great. So we are at the end of our time. If you have any additional questions, feel free to go ahead and submit them. If we did not get to your questions, we'll follow up with you by email. Now, I would like to thank our speaker, Dr. Beate Litzenburger, for joining us today. Thank you also to the audience for your attention. We hope that this was informative and helpful. So this concludes today's session on the Human Somatic Mutation Database, or HSMD. And we look forward to hosting you in a future QIAGEN Digital Insights webinar. Thank you again, and I hope you have a great rest of your day.